Hey everybody, it's Merritt. I am filling in for Dr. Ramson, and I'm a professor in biomedical science as well. And my work is on neurogenesis and in neurodegeneration. So it's a fun thing to talk about learning and memory with you guys, which was, I know, up for discussion as you guys are looking ahead from neurogenesis. So what I thought I would do is link some of the things that happen during early embryogenesis and how the nervous system's set up, and then take it from there. So we'll cover some of these things online, and then other things we'll actually cover in class on Wednesday when we meet. So, sorry, first slide. Just a stupid pun, but basically that the brain is plastic, but not made of plastic. And then a little bit about some of the basics of like how do you build a circuit and remodel them. So how is it that we actually make, we've talked about, or you guys have talked about neurogenesis and how these neurons are actually born. But then once they're born, how do they get to where they need to be? How do you set up and establish these types of circuits? How can they change in dynamic environments, including in learning and memory? And the next phase of it and that next set of slides, what we'll cover mostly on Wednesday, is about memory and amnesia. And we'll talk about structures and different types of memory, sort of a much higher level analysis. But here, we want to kind of get an idea of how these things are being laid out and how they work. So what I wanted to do is basically show you guys an outline that we have here. So what we're focusing on in this particular picture is where we have the visual system. So this is a mouse brain or a rat brain. And back in the occipital lobe, which is here in the mouse, this area 17, which will be familiar if you've learned some of the Brodmann's areas, but if not, then it's one of the primary areas that we have for the visual cortex. And in that area, this region that they describe as being the striate cortex here receives a lot of inputs from the right and left retina. So these circles here are depicting what the retinas are actually receiving in terms of input in their visual fields. And if we look, the retinas receive input, and let's say that it strikes upon a photoreceptor in this blue area here. That gets carried back to, from the photoreceptor, it gets transduced through, guys, if you cover that in a visual component, you'll learn some of the steps. But the main idea is that this leads to forming part of the optic nerve, one axon, that then feeds back to the thalamus. And then from the thalamus, then it feeds back to the striate cortex. And likewise, the same thing happens to the nerve on this side. And so these both end up converging onto the striate cortex. So a lot of these details about, how the photoreceptor works or how the wiring works from one cell to the next, etc., are outside of the scope of this particular conversation. But the thing that we want to be able to appreciate is that there's two different eyes carrying information back, representing the same part of the visual field. And this input that feeds back to our striate cortex is monitored. So what you basically have is two representations of the same visual field, but from slightly different angles, because one is coming from one eye and the other one's coming from the other eye. And so it's represented and passed back to the different levels of the cortex. And so here we have layer 4 of the cortex, which is where you receive input from sensory inputs into the cortical areas from the thalamus, like what we have from the part of the thalamus that carries visual input. And so those layer 4 neurons then pass their information on to layers 3 and 2. And what we'll see here, and we'll get into the weeds a little bit about, is thinking about how these two different cells from layer 4 actually end up communicating with the ones in layer 3 and 2. Because, as you can see, sometimes there's overlap. They talk to the same types of cells, and they give different types of input to them. And this is important because of the ability to actually be able to see in stereo. In other words, to be able to have stereoscopic type of vision, to be able to discriminate and have depth perception, you need to be able to have input from both eyes. If you've ever tried to drive or move with just one eye open, then you realize that you've lost depth perception. So this ability to be able to perceive depth perception relies on this kind of wiring that's established here. And it's a really nice type of input because there's a couple of things that happen here. This type of depth perception is important for us being able to, first of all, set up wiring. It's amenable to being changed, so it's plastic is the term they used to describe it, and hence the pun I had there about the brain being able to change, being referring to it as plastic. In the opening part of the title of the lecture. And then we have this, and then not only that, it can be distorted by pathology. And finally, it helps to represent the type of changes that can happen within these cells that ultimately can also give us insight into how learning and memory occurs. So this is all about sort of setting up things and then remodeling and changing them so that the brain can be able to respond to the dynamic environment into which it has to operate. So let's go ahead and take a look then as we kind of dig into this. So there's basically, the first thing we're talking about is sort of how these neurons actually set up their paths. So there's a pathway, a target, and a so-called address. And the main idea out of this is showing that we have this trajectory of a growing axon that then has to choose whether it's actually going to cross over the octave chiasm here and continue onward, and where it's going to actually innervate in different parts of the thalamus. And so these are kind of illustrated here in these different points here. Point 1, the octave chiasm, 2 at the thalamus itself, and then finally then these thalamic neurons then go through a similar process connecting up to the cortex. So we have this kind of path that's being shown here, and then a target, a rough, like the thalamic area, and then the particular address, which is really specifying where it's actually going to. And so we'll see a little video of this that I think will make it actually seem a little more dynamic and interesting. But right now we have this kind of splat looking like picture here. What it's trying to depict and show here is in the growing axon that we have these lamellipodia that stick out from the extension of the neuron. So we have these new neurons that you've learned about that have been generated. Now they're extending out their axons and need to set up the appropriate wiring. And so it's all about how these different actin filaments within the lamellipodia and the filopodia, for that matter, are able to actually assemble. If they are able to be more rigid and are able to polymerize more readily, then they'll extend out and they'll continue to build. And this particular filopodia, for example, will gain a longer length and will become a dominant one. And so anything that can help stabilize the actin filaments, this is really dynamic in here, will be important for being able to help promote growth in one direction versus another. So it's really kind of a cool dynamic little system here. And these filopodia basically are these spikes that project out outwards, and they're dynamically regulated. And when they adhere to this extracellular matrix, they can carry that forward. So let's go ahead and just take a look then at some of the sort of schematic depictions of how this works here. So what this is actually showing is multiple different cells. So, for example, this cell right here has all the lamellipodia and all of those types of things adhering to the extracellular matrix, which is shown and depicted here with these little, we have the purple points here that are depicting part of the laminin molecules present in the extracellular matrix. We have integrin molecules that are presented by the axon that then connect to the laminin molecules and ultimately allowing adherence to occur. And so that's just one of these guys that we're talking about. So here's the axon, and then here's where it's sticking. And so some of these filopodia are getting some traction, and that's what's being depicted here. The other part of this story that they want us to be able to say is that we have these cell adhesion molecules that allow cell-to-cell adhesion to occur, and this kind of fascicles to be able to form. So if you've heard of fascicles in terms of anatomy, describing sort of bundles of axons together, that's what we're seeing here. So now we have these cell adhesion molecules, or CAMs, being able to link all of these different axons together. So they're following along with this axon here. So these help to coordinate the projection of multiple different axons along this sort of highway, if you will, the molecular highway as they describe it. And this helps to direct and cause the connections to actually occur appropriately. So we have this kind of, and when we think about these genesis of connections that's actually happening here, we have axon guidance, and this has a couple of different points to it. We have challenge. The challenge is basically wiring the brain, which is a big one. And some of those challenges include being distances between these different types of connected structures. So it's pretty easy in the early stages of development to be able to establish these types of major connections. In the mature nervous system, that's a much bigger deal. So for example, being able to actually intervene with a type of therapy in the adult can sometimes be limited in terms of trying to set up wiring from distant parts, like for example from the substantia nigra where Parkinson's has a big impact, all the way to the striatum where they actually synapse. That's not easy to reestablish in the adult and hasn't been done so far. They've had to try to do other workarounds. But in the early nervous system, these are really close to each other, and so it's easier for the axons to actually make the connections. And these pioneer axons are the ones that actually stretch and help guide neighbor actions to the same target, much like we're seeing here with these pioneer axons acting there. And then this allows growth to occur in the correct direction. But when we think about how axon guidance works, it's kind of interesting. Like, how does this process, how do they know where to go? And the answer is basically that they have these axons, as they project along, have guidance cues. So these guidance cues interact with growth cones that are present on the actual developing the neurons here. And what's being shown in this particular picture is a cell body here, an axon projecting outward, growth cones that's representing this big structure basically here, and then all these little filopodia. But what's been added to this particular picture is that we have netrin receptors that are sort of integrated within this. And it's illustrating one of a particular type of cell, of receptor that we actually have. So netrin receptors are ones that will help to polymerize or cause the melopodia and the filopodia to be able to extend outward. So actin will polymerize more readily and stabilize more readily, and you'll have it drawn towards a particular direction. And the netrin substrate, or rather the ligand for the netrin receptor, is released in diffusion gradient from a particular area here. So this kind of blob that's being depicted here is actually trying to represent the spinal cord. And in the spinal cord, along the bottom part of it, or the ventral part, meaning sort of the part that's facing to the interior of the body, what we have is a really high concentration of these netrin molecules that then are actually being able to diffuse outwards towards this particular structure here. And so it's being drawn towards this particular area. Great. All right, cool. So what? So there's also, though, another type of receptor that's known as the ROBO, which is present on these neurons as well. And so these ROBO types of receptors are sensitive to another ligand known as SLIT. So what happens is that netrin receptors are expressed here predominantly in this early stage. So it's kind of being drawn into here. And then what happens is that as it projects outwards and beyond here and moves out beyond this midline, the cells change. And they start to express ROBO. And ROBO, what that does is that actually is a chemorepellent. And so the further you get away from this, SLIT, which is this chemorepellent here that interacts with the ROBO receptor, that is what the better off and more stability that you're going to have. So to restate that, just to make sure we're clear, this I think is easy, right? Netrin's here, we're all being drawn to it. More polymerization of actin being drawn to that area. Great. As you cross the midline, cells change their receptors. They swap out and they start expressing ROBO. ROBO's picking up this particular ligand that's being released here in a diffusion gradient that's called SLIT. That's the ligand itself. It's being sort of diffused outwards. SLIT acts differently. It's a chemorepellent. Higher concentrations of SLIT would make this collapse. And so the cell is drawn to moving away from that area where they have less SLIT, and so it continues onward to actually reach its destination. Because this particular neuron, its goal, or the way it's typically set up, is that it will go over and cross the midline and reach the other side. So let's take a look at this video that I've added a link here. I have it also present. I'm going to see if I can get this to work with me. And there we are. Okay, so here we are. Take a look at this cell here. This is the dynamic neuron that you can see. It has multiple different, here's a growth cone, lots of filopodia coming in and out. Once you add SLIT, the chemorepellent, look at how these filopodia start to shrivel up. They bathe the entire neuron in that, so it's just going completely away. So just to see it again, just because it's super cool, and it helps us to illustrate it, look at all of those different polymerizations of actins that's manifesting as these lamellipodia. You add your SLIT, now you cannot have, it's interesting, it makes these longer connections there, but ultimately the impact is that there's depolymerization of the actin, and the growth cone collapses. So that's what's happening. If this neuron stayed here, it would end up collapsing. But what it does is that it starts to grow towards where it has less of the SLIT present, and it polymerizes in that direction, ultimately then moving away. So kind of a cool way that these genesis of connections actually occur. So when we look at how this actually works then, we see axon guidance, what's going on in this particular case. So this helps to establish these topographic maps. And I'm going to sort of ask you to think back to originally what started our conversation on this was that we were talking about how the visual system's actually hooked up, and that we have inputs from the thalamus that go up to the cortex. And we can see that here. And it's all, we have this wiring that goes up to the cortex here and innervates layer 4 of the cortical layer 4. And then those in turn innervate layers 3 and 2 that are getting inputs. So these end up being topographical maps. This little patch right here is represented in one area of the striate cortex. In other areas of the striate cortex, we have other representations of the retina. So these ultimately end up being topographical maps. So they're set up. We have these different types of ways that they're established. First is that we have these retinal axons that innervate these targets, like part of the thalamus called the lateral geniculate nucleus and the superior colliculus. And then early in the 40s, this chemoaffinity hypothesis, in which you saw some of that come forward in the SLIT and ROBO, but we actually had much earlier iteration back in the 40s with frog work, was that helped to establish and how this kind of wiring was actually done. And so, what we were looking for is just different factors that were helping to guide these different neurons. So unlike the spinal cord, in this case, we're looking at efferents, or the ones that were important for the retina. The major thing I wanted you to be able to come away with, though, is that we were able to establish these kind of topographical maps. Chemoaffinity is an important part of it. And in the retina, we were talking about how these things actually end up, wiring out that way. So let's talk a little bit about how these different synapses are made. And a lot of it has been done at the neuromuscular junction, and then ideas have been sort of adapted from the neuromuscular junction to how different neurons may communicate with each other as well. And part of that is because the neuromuscular junction is really easy to map and really easy to get to. It has a lot of technical advantages to it. But if this machine cooperates with it, I can actually show you some of the newest data that they have on those types of connections as well. So let's go through some of the basics that we have here. So here's a motor neuron, and it has here's with a growth cone and an axon terminal, and then it's interacting with the basal lamina of the extracellular matrix, and then here's our plasma membrane of the sarcolemma, of the muscle cell itself. So what's going on? So we have a couple of things that are happening. First of all, there's a signal that's being sent from the motor neuron to the muscle cell called agrin. Agrin binds to a receptor here called the agrin receptor, or MuSK here. And what this does is that this helps to activate this rhabsin molecule here that ultimately then helps to anchor and aggregate, hence the term agrin, these acetylcholine receptors. So you may remember from other classes that the acetylcholine receptor that's present in the neuromuscular junction is critical for being able to pass information from the motor neuron onto and depolarizing the muscle cell itself. So acetylcholine is released from the motor neuron and then binds to these nicotinic acetylcholine receptors that ultimately allow sodium to come in. Well, in order to set all that up, you have to have this kind of a process occur. So identifying these molecules was a big breakthrough in terms of seeing how it was that a motor neuron could actually cause this aggregation of these acetylcholine receptors that ultimately clustered and then formed these invaginations that we see in these junctional folds that appear within the neuromuscular junction. So the same kind of idea is thought to happen in the central nervous system as well. In this case, we have a growing axon, and then we have a dendrite of another neuron. So the axon and then its potential connection. So remember, axons are sort of the output part of a neuron, and dendrites are where you're getting your input. It's where another neuron's getting input. And so what's occurring in this particular case at this stage is that there's a filipodium that's being drawn towards this growing axon, sort of like what we might have seen between a chemical affinity between the motor, this axon here, and the muscle cell. And this filipodium reaches out and then is able to help establish a connection. Synaptic vesicles are present, and a presynaptic active zone is seen. After this active zone is set up, then the next step that occurs is that it helps to aggregate and cause a cluster of postsynaptic receptors here on the dendrite. So this happens in the early stages of development, but this can happen later as well. So a really kind of cool kind of thing that we can think about then is how is it that these neurons are actually helping to establish and set up connections within themselves and sorting themselves out during development. So one of the things that we have to do is once we make these types of connections, some of them are not going to be appropriate. Early in development, we make more connections than we keep. So we have to eliminate some of them. And so the simple mechanism, there's a lot of text here, but let's just focus right here on the picture and then what happens next. So main idea is that early in development, you have, or the initial state is described here, lots of input neurons hitting multiple different target neurons or muscle cells, whichever system you're looking at. Afterwards, after development proceeds, some of these connections are favored and others are not. And the ones that are favored will not die, and the other ones are pruned back or undergo cell death. And so what you can see here is that there's these single connections that we have between them. And what's thought is that what causes this cell death is that there's competition for growth factors or trophic factors that are released by these target neurons. And so if this one happens to be close enough to actually hit this particular target neuron, then it will live. Whereas this one is too far away and not able to reach that particular neuron, and therefore isn't able to get sufficient trophic factors to be able to help it. So this is more than just a hypothesis. They've actually tried to eliminate these different potential trophic factors, seen this kind of event occur, and so it's been tested as one of the mechanisms by which you actually see this occur during development. So these mechanisms of pathway formation that we have allow this kind of large-scale reduction in the number of neurons and synapses that we have. And ultimately then, in the development of brain function, we're looking at this balance between making new neurons and then eliminating them. And so cell death is one of those ways that's manifested. So, all right, cool. So then let's take a look at what happens in this type of an example of these types of elimination that occurs here. So in synapse elimination, what happens in one particular case is we're looking at how alpha motor neurons connect with muscle cells. And again, we're choosing muscle cells because they're easy to look at, they're nice to characterize, and it's a pretty robust and well-defined system. But they are doing this type of work also in the central nervous system and trying to pull apart these micro circuits that we have as well and how development interplays with them. But right now, these are some of the better, the ones we had the best insight on. So here we have an alpha motor neuron that's then innervating with multiple different muscle cells here. So this polyneuronal innervation of these immature muscles. And then what happens over time as you have maturation occur is that these neurons then pick a couple of, or pick one of the particular neurons. They have multiple connections with it, but just one particular muscle fiber that it actually innervates. So one neuron innervating one particular muscle fiber. So they've done some really cool experiments back in the day looking at how if you blocked activity out of these cells, so acetylcholine receptors, if you block those acetylcholine receptors in a small area of the neuromuscular junction, what ends up occurring is that you end up getting remodeling occurring. So even though these things are set up in adulthood, here, here, and here, it's not like that's set in stone. If you block part of it, then what will happen is that you will lose that portion of the neuron here, and the neuron will eventually just prefer the parts that it actually has an active connection with. So there's a withdrawal of the axon branch at that particular site. So if you don't use it, you lose it. So that's the kind of thing that happens with this kind of wiring. What I wanted to show you and is this particular video that we have that illustrates how how these synapses are actually modulated. So let me just see if this will connect for us. Let's use this one. Let me see if I can actually control it. All right, I'm going to try. Going into. Sorry about this. Let me try again. Okay.
We were motivated to do this study by a very obvious fact that anyone who's seen a baby can appreciate, and that is that young mammals, humans especially, behave very differently than adults. We had some hints from work that we and others had done in late stages of the development of muscle that there is a certain amount of extra wires, like double the number of wires one sees in the adult in young animals at about postnatal day 7 in a mouse. And that work had been published quite a while ago, and we were wondering whether that's the extent of the extra wiring, or if we looked earlier in development, might we see an even more profound change. We really didn't know. And
so they go through this whole process, and
we couldn't answer this question because technically we didn't have.
this is going to take too long to get to the part that's really cool. So I'm going to have to end up posting this for you guys. I'm sorry. But there's what's really nice about that study, and I'll share that video with you where you can take a look at it. But the idea of it is basically showing that there's they've gone through and done this new technique of basically serial electron microscopy, now available to do because they have the processing power to be able to computationally stitch together all of the different electron microscopy images that they're able to get from electron microscopy, and then be able to map all of the different connections that they have. And they're basically showing that this kind of a picture is really kind of true and that there's multiple connections with it. So it's really beautiful and interesting, but I'm afraid it's going to take a little too long to get to that main point with the control that I have in there, so I apologize for that. All right, so let's get into this. So we have synaptic-dependent, activity-dependent synaptic rearrangement. So what's happening here? So the main idea is basically that fine, we've set up these new neurons, or we've gone through, we've made the new neurons, they've wired up the right way, and now they're in a position where they can actually respond to their environment. How do we have this kind of arrangement and rearrangement occur? And how is this actually kind of in a meaningful way? And so looking at this early stage of development is actually really cool, because right after birth, you can have this kind of dramatic types of shapes that reshaping of the cortex that can occur. And they did it in cats. They did a lot of work in cats because, as you know, cats will wake up, were born with their eyes closed. And then when they open, then the light stimulus is able to come in, and they're able to actually, process and be able to establish better connections within their visual cortex to be able to keep track of and develop stereoscopic vision. And so what we're going to be doing is looking at sort of images that they were able to collect from this. Hubel and Wiesel did these in classic studies back in, I believe, the 70s. But it's still really powerful for even today. So presynaptic neurons A and B here, for example, are targeting a particular cell. And synaptic rearrangement comes into play where, in particular, maybe B has a much more kind of potent connection with the target cell than A does. This can happen in multiple different ways. This kind of synaptic rearrangement, for example, could happen because B is more in tandem with firing with the target cell, and that can help promote synaptic connection. And this can be out of phase with a particular target cell and make it so that it's not firing as readily. And this sort of idea of being in phase or out of phase sounds a little bit spooky, but it kind of makes sense. If one cell is better at being able to sort of hit this particular target cell in a meaningful way, then it's able to help. Then that type of synapse is actually favored. So we'll look at some examples of that as we consider this. So fine. Synaptic segregation is this idea that where we have a refinement of synaptic connections that are occurring. And there's a couple of things that are out there that help to do this kind of segregation. And we're going to be looking at sort of how the retina talks to the lateral gingulate nucleus. And then ultimately we're going to talk about how the LGN talks to the striate cortex, wherever the visual cortex is. So we have these retinal waves, which are sort of these activity that's happening within the retina of the developing embryo before it actually opens its eyes. And so it's relatively uncoordinated, but there is activity there that helps to establish the types of connections there. So your eyes are closed because you haven't opened them yet early in birth, but there's still this kind of like waves of activities that sort of course across your visual cortex. And then there are these ideas of the Hebbian synapse that help us to be able to specialize or favor one particular type of a connection than another. So let's take a peek at what we're going to be talking about. So one thing is that we're going to be looking at how the retina goes from here and then connects to the thalamus over here. And so this is a much more detailed picture kind of showing how one particular part of a visual cortex or of the retinal field, like let's say over here and over here, picking up maybe a visual stimulus from up here that happens to hit here and there. Or maybe, I'm sorry, maybe, visual stimulus right up here, hitting here and here, for example. And then that representation is then carried over to the other side of the brain, other side of the midline. So there's eyeball 1, eyeball 2, there's your nose, and something over here is ultimately going to be projected and processed on the opposite side of the midline in your visual cortex. Retinal ganglion goes back, shoots back to the thalamus, and we see the same thing happen over here. Awesome, great. So there's multiple different layers of the thalamus that it's outside the scope of this conversation. But the thalamus then ultimately projects back to the visual cortex. But within the thalamus, we actually have dominance in terms of whether they're coming in from the right eye or the other eye. So you can see these dotted lines, for example, are carrying information from the contralateral eye and innervating layers 1 here, layer 4, and layer 6. So whereas these inputs coming in from the ipsilateral eye are coming in and they're innervating at layer 2 and layer 5, and that's it. I'm sorry, layer 3. So 2, 3, and 5. So these are the ones where you're seeing it kind of come in and feed into to that side. So this kind of specificity is really interesting. And so how is this kind of specificity actually established? And what happens essentially is that, and we can see this kind of one eye being shown going to this layer, another eye coming into this layer. That's the depiction that's being shown there. So the principle here is basically that there's plasticity at these, what they call Hebbian synapses. And so what's occurring is basically that as these connections are made, they're favored more readily in one particular type of retina and how the retina connects to the thalamus here than it is on this side. So more connections are being made to this particular neuron. More of these axons are projecting and hitting this particular one. So so that means that when this target is activated, let's say by this guy right here, and then this one happens to fire in synchrony with it, then they're going to coordinate, and there's going to be a favored type of connection between these two. And there are factors that are responsible for when this is firing and when the target cell is firing at the same time, and there's synchrony, then they end up being able to establish their connections more strongly. So it's the idea that cells that end up firing together, wire together, is the principle here. And so from the other eye, these tend up, if you remember, we had those pioneer axons that were first setting up their connections, and then we had others that were following along through cell adhesion molecules. Well, now they've kind of, ended up at one particular spot or another. And there may be some messiness here, but that sorts out pretty quickly because a majority of the neurons are connecting with this target cell here versus the ones over here. And so that means that the activity out of one eye is going to be mostly correlated with activity out of this target cell because that's the predominant amount of input that it gets. So anything that's out of sync, like input from the other eye, is going to end up getting eliminated. So let's take another picture of this and look at what happens when we go from the thalamus up to the primary visual cortex. And so we're going to be looking at these what are known as ocular dominance columns. So we have in the upper level of cortex, or in the cortex, we have these multiple different layers, and these layers have different types of functions that are associated with processing visual input. So our focus and the pictures we're going to show are basically sort of a cross-section of looking from most superficial to deeper layers of this primary visual cortex and the type of input it gets. So in this picture, what we're seeing is a depiction of layer 3 here, and then layer 4C, which is where a lot of the input's coming in from the thalamus and going into the cortex. So what's the purpose of this particular diagram? What do we need to get out of it? The thing that this is trying to represent is much of like what we saw in the very beginning. So I'm going to show you that early slide just to make us sure that we're making good connections here. If you remember, we talked at the very beginning how layer 4 connects with layer 3 and how there are these types of connections that happen here. And I described how the right retina and the left retina are carrying in information. Well, now we can see that these types of connections are being, we're investigating these types of connections. So the schematic that we're looking at is inputs that are coming in from the thalamus and feeding into the cortex, and then we're looking at how these wire up. And there's dominance that ends up occurring with some of these layer 3 cells. So now let's go back to our slide that we were at and take a look at what's going on. So what this is trying to depict, where we have red and blue circles here, are trying to depict cells within layers 2 and 3. And they're just showing out of that layer 2, in that particular cell, what's happening. Is it binocular or monocular? If it's monocular, it's going to have a blue or a single color, like blue or red, representing either the ipsilateral or contralateral eye. And then if it's binocular, then it's getting input from both. And what this is trying to show is that input coming in from the thalamus into layer 4, and then cells in layer 4 ultimately then projecting up to one of these, into these cells up in layer 3. So monocular or binocular. Okay? So that's what we're looking at here. So there's some overlap here between two sides, and so you have a binocular input right there. Having binocularity is really important. Having monocularity is really important in terms of being able to have stereoscopic vision. So animals that have strabismus, or either induced or otherwise, strabismus is where you have basically a lazy eye, or there's some kind of ocular muscle issue that's occurred. Then your number of binocular inputs is distorted and compromised because there's not an alignment of an input between the left and the right eye enough that they actually end up wiring together. So what these striations are trying to show here, this picture is showing activity that happens within layers 3 and 4 this is layer 4 out of the cortex. So these are inputs that are coming in from either the left side or the right side of the eye. And so if you block input into the developing retina, so the retina is not able to fire as much, you lose these stripes. So what are the stripes representing? The stripes are representing, and this is the blue line trying to show sort of the cutaway here, we're looking down on layer 4. And what it is that the light parts are showing where there was activity that's occurred in a particular cell. And what they did is that they have one eye that's open and one eye that's shut. And so they can easily map which that this is from the eye that was open, and the eye that was shut is over here. If you blocked activity in the developing retina, you don't see these types of differences as readily. There's not as much striations and as you might expect. There's a tremendous amount of overlap that can occur between these two things. And so without this kind of definition, it can be very difficult to be able to have appropriate stereoscopic vision. So this type of plasticity doesn't last forever. And so there's a critical period which is important for this to actually happen. So what we'll see then is that this critical period is happening during development, and it reflects this kind of plasticity of these ocular dominance columns. So the way they tested this, as I mentioned before, is that they did this what they call monocular deprivation and knocked out one eye, and then saw these types of striations and other types of things occur. So let's take a look just at the quick list of what they did. So they looked at the plasticity of these different ocular dominance columns. What they found is that these stripes that were occurring here, if you closed one eye and left the other one open, then one particular eye would dominate. If you blocked both eyes, then you would lose the ability, you would lose this, like what you see here, where you've lost the type of specificity of input from one side to the other. So those are two examples of what what they were able to do in here. So how is it then that you establish this binocular vision up here in layer 3? That we have this kind of input, ocular dominance. We talked about ocular dominance happening here in layer 4, but then we have binocularity occurring up here on layer 3. So how does this binocular part happen in layer 3? So if you measure up there, what they did is they made a graph. And what this graph is doing is that they basically stuck in an electrode and monitored. And they stuck an electrode and they weren't really sure where it was going. They would just monitor, they'd track it, see, okay, looks like we're hearing some activity as they're moving the electrode into the visual cortex. And then they would shine a light into the eye of the cat, one side or the other, and then monitor to see what would happen to its function, like, you know, whether it was firing a lot or not, whether it was monocular or binocular, etc. And so what they found basically was that there was, out of the neurons that they were measuring in layer 3, that, you know, there was about this number here, about 35% of them were actually binocular. And then you can see that it kind of, that dominance varied here, and they roughly categorized them into 5 different groups, where there was strongly monocular, either contralateral or ipsilateral. And then you had the middle, which was where they're almost perfectly binocular. So then what happens then if you did monocular deprivation? So think about what you think would happen in this particular case if you had monocular deprivation of these eyes. And what is happening, and what you, so, you know, kind of give yourself what you think would happen. So let's say that you blocked the contralateral eye. It was a closed eye. Then what's going to happen to this distribution? Those cells in layer 3, are they still going to be binocular like they were before? That would indicate that things were set up to wire, and that if you did monocular deprivation, it shouldn't change it at all during that critical period. So this is early development types of monocular deprivation. So, but if you don't see this preserved, then that suggests that there's an activity-dependent type of process or a dynamic process where if you're receiving only one type of input, you'll get one type of response, and that's what actually ends up happening. Most all of your eye is in the open eye here, and this. So what they do in this is they close one eye, and then when they actually do the experiment, they open the eye back up, and then they monitor to see what's going on in terms of activity out of the cells that we have dedicated in layer 3. And so from the closed eye, there's almost no representation of activity from there. There's a little bit, but not much. So, so the favor has gone to the most active pathway that you have. So there's a couple of kind of ideas that happen here. And one of them is this idea of what they call synaptic convergence. And we talked about monocular deprivation, but the other thing I want to talk about is this kind of convergence that happens by having both inputs happening at the same place. So let's look at this particular type of example. In this case, what they've done is that they have, here's our normal type of situation in layer 3. So, but what they've done is they've done some kind of intervention here to affect the animal. And the question to kind of consider here is, looking at this, what do you think happened? Why is it that you have all monocular here and here? What could actually cause that to occur? So the answer is that what we're looking at is these animals were imposed with strabismus. So they had part of their ocular muscles were cut so that they were, cross-eyed, essentially, in one way or another. And that meant that they were never looking at the same spot with both eyes. So activity from both eyes would never reach the same neuron. You never have the opportunity for binocularity because the convergence never was there in terms of activity. So let's go back to this. your left eye input and your right eye input, in order to have a binocular input, they have to fire together in order to wire together. If your right eye is looking way out in the different direction than the left eye because of some kind of strabismus, they'll never fire together because they're going to be looking at different parts of the visual field. Whereas if they're both attuned to the same particular visual input and they're getting a light flash or something like that, there's going to be synchrony that's going to feed up to this layer 3 cell. So that's what we're considering, and that's what's happening in this particular example, where there's asynchrony. There's not a type of, they're not able to connect. So they're out of sync with each other. And the maximum is basically if it's out of sync, then you lose your link. So kind of funny. So here we go with the kind of rules that we have then for synaptic modification, which is basically that the cells that wire together, fire together, and that the ones that are out of sync, they end up losing their link. So, okay. Now we've learned about this type of thing. Meaning how activity, how these different neurons are connected and so on. So what we want to be able to do is to start to think about how this might relate to memory. We've thought, in other words, we've made a bridge. We've talked about new neurons being made. That's what you guys talked about before. How these neurons actually map out, move across, set up some of their wiring, how that wiring depends on activity, how that activity needs to be correlated in order for it to actually happen. And so one of the most powerful things that you guys will be learning about is long-term potentiation. And so what happens in this particular type of circumstance is basically that there are two different types of these LTP receptors here. So we have they're both related to glutamate. They both respond to glutamate. One is called the AMPA receptor, another one's called the NMDA receptor. And there's complexity here that I'm not going to get into in a lot of detail. But what is occurring basically is that glutamate is released, and under normal circumstances, glutamate will activate the AMPA receptor, but isn't sufficient to actually unplug the magnesium block that's present on the NMDA receptor. That's at rest. And what essentially happens is that when there's a lot of depolarization of this neuron here, let's say this neuron here, the positivity that builds up in here because of the AMPA receptor being active and other things that might be activated, what ultimately occurs is that it helps to push out or repel that magnesium plug. And now glutamate can not only activate the AMPA receptor, but also this NMDA receptor. And the really cool part about this is that it allows for what they describe as sort of a molecular coincidence detector. Because what's really special about this NMDA receptor is that it allows calcium to enter into the cell, and calcium is a crazy robust signal for being able to induce types of changes downstream that ultimately can have profound impacts on how the cell behaves and responds to its environment. So it's an incredibly strong signal. And so what this is basically saying is you get a calcium burst when there's a lot of depolarization from the neuron that is connecting to this neuron. So when this presynaptic neuron is dumping a ton of glutamate, activating a lot of AMPA, then it's also going to be, it's going to depolarize enough to pop off that magnesium block, allow calcium to come in. And that actually promotes and allows remodeling to occur between the two synapses there, and ultimately is responsible for some of that synaptic activity dependent coordination that we talked about before. So there's a lot of types of ways that this can work, but let's take a look then at how this kind of plays out. So this is sort of the list of things we'll get into, but let's look at the diagram, and I think that will help us get into what we need to. So what we're looking at is multiple axons that are projecting onto the dendrite of a particular neuron. And we're just measuring what's happening in this particular neuron here as a postsynaptic EPSP. which is an it's an excitatory postsynaptic potential. So we can see it's kind of excitatory, depolarizes, and comes back down. We're going to zero in on this part. Here, before LTP, here, during LTP, and then here, after LTP. Here's the dendrite, and there are the axons. So here and here. So what happens? We're also looking at, in this place, NMDA receptors in red, AMPA receptors in blue. And you will remember these players from what we saw here. NMDA receptors here, shown also in red, and AMPA receptors in blue. So what happens before LTP is basically that you have firing that occurs, and there's a bias here. And what we're seeing is that there's more connectivity, or at least more synapses being shown out of this particular vesicle, or this particular terminal, and this particular presynaptic bouton on the dendrite. So during LTP induction, what happens is that there's enough depolarization out of all of these, this one, and this one, that they all induce the NMDA receptor to allow calcium to come in to that mechanism I was describing before. So calcium comes flowing in, and that causes these remodeling changes to occur, what they describe as amplification. The AMPA receptors here get shuttled up and inserted into the presynaptic membrane, and now it's much more sensitive here to the input that it had before. So now we see that there's more blue up here and here and here than there was before. So now the connection between when this neuron, this axon happens to fire again, it's much more likely to actually cause depolarization to occur here. It has been sensitized to that particular type of input. And so now we can actually get a much more robust type of firing. So this is that firing together, wiring together in some ways, because they were all activated at the same time. They kind of show this as where all three of these are being activated at the same time with a little shocker. Because they're being activated at the same time, they're able to cause depolarization more readily at the same time as well. And you get the same type of connection that's occurring for all of them. So that's the kind of long-term potentiation that can happen. And they describe it as potentiation because then it can last for minutes, even days afterwards, where beforehand it might have been like at this level, they call this 100% sort of baseline here. And then afterwards, when you zap it again, they have a much higher amount of their EPSP. And this is just a rough estimate of how fast this went up. It's the slope of the EPSP. So it's showing that the slope went up really, really steeply, and you had a much more robust type of of depolarization there. Okay, so then there's also long-term synaptic depression, which is sort of the opposite of what we learned about for long-term potentiation. So in this case, what we're looking at is that when neurons are firing out of sync, so unlike here where they're all firing together and then you get this kind of depolarization and everybody's getting psyched, here what's happening is that these neurons are not firing together. And so what happens is, we saw monocular deprivation occur and the profound effects of that and binocularity that happened in the striate cortex back when we did this particular type of test, right? That was during the critical period of development, so really profound effects. But this is in adulthood. This is when everything's set up. It's after the critical period. And what they did in this case is that they took and they monitored the activity of these visual cortex, number of action potentials from the left eye and from the right eye. And then they did 17 hours of right eye deprivation. So not even a full day, just one eye. And then they tested to see how these neurons would fire. And you can see that the distribution and sensitivity of the striate cortex has shifted over to the left eye. So this tells you how dynamic and how sensitive we are in adulthood to this type of difference that we have within our visual cortex. A profound illustration of how plastic our brains really are, how we can respond to our dynamic environment, how we encode that. So here we have long-term depression, And what we're looking at is, and we're seeing that these neurons are firing out of sync. So the left eye and right eye used to be a binocular situation. Used to be one of those cells that was hooked up, much like we see here. And if we were to go back to the picture that we saw, something where the left eye and the right eye were coming in and firing together. But now, you take away input from one of those eyes for 17 hours, and then it doesn't look like this anymore. It's changed, and so it's much more monocular than it was before. Okay. So that's what we're seeing in this kind of type of case. So how does this occur? So the idea basically is that when you have weakly correlated activity, then the type of connections that you would have between the dendrite here and the presynaptic neuron here, where you have the axon that's releasing the synaptic vesicles here, if they aren't correlated together, then what will happen is that there's the AMPA receptors end up getting internalized and getting taken in. So there's a loss of these particular types of receptors. And they end up being recruited for something else, where there's actually, and taken in and used for other, the pool of internalized AMPA receptors are then utilized for where there is activity from the open eye. So this is activity coming from the closed eye here, and this is activity coming from the open eye over here. And so this gets sensitized and this gets downregulated. So to kind of illustrate this, they're showing an axon in this particular case from the thalamus feeding into the cortex here, like this guy right here. And it's firing kind of randomly because we've shut that eye and we've like taped it shut or whatever. And so this one's not firing quite as strongly either, and this one isn't quite firing as strongly either. And so there's not a lot of coordinated firing here. Shown in blue, it's kind of out of sync. And there's just not a lot of stimulus there. And so the neuron's not wasting any time by just remodeling, taking away these receptors here and putting them over to where there's nice correlated patterns over from the eye that is left open. So that's what's occurring in this type of case. So we're seeing this kind of listed out here where we have this sort of the opposite of LTP, where we lose synaptic AMPA receptors here. And, there could be a potential loss of synapses as well, although the time course is pretty fast for that. So there's also, you might have noticed this, that there's changes that can occur, especially like when we looked at LTP back here, where the number of vesicles here is different than the number of vesicles here. And that schematic is actually hinting at something really interesting, which is basically that there are presynaptic changes that occur. In other words, it's not just unidirectional where it goes from here over to here and causing a change to happen on the postsynaptic side. There's this retrograde signal that goes back and helps remodel this. Some of this is related to the action of nitric oxide, and others is related to the action of cannabinoids. So we're going to stop there. When we pick up on Wednesday, we'll talk about temporal lobes and declarative memory. And this will help us lead into memory and amnesia and talking about some of those different features there. We'll get more structural in terms of our conversations about this, a little less molecular and a little less about how these cells are wiring together. But I hope this has been an interesting ride.